Greetings, biology students. This video is designed to help you complete your very first lab report. So our lab consisted of observing through the microscope plant cells and animal cells, human cells. We actually took a look at the human cheek cells. So let's talk about how you can successfully complete your lab report. So first things first, we have to have a title. So each lab report should have a descriptive title. The title should clearly explain what you are studying. Also include your name and the date of the laboratory experiment or observation. So our lab was conducted today, September 19th, 2024. When we say that your lab should have a descriptive title, it may be entitled Observation of Plant Cells versus Human Cheek Cells. That is a very descriptive title. It gives the reader an idea of what you're doing in this particular lab. What you should not include or should not entitle your lab is, hey bro, we used the microscope to check out these slides. Right? That's not going to be professional, and please don't have the name of your or the title of your lab report to be something along those lines. Okay, let's go to the next section of your lab report. So the introduction. So in our laboratory that we performed today, it was mainly an observation lab. So we don't necessarily have a problem that we're trying to solve. But what you should state is, we studied plant cells, and I understand that the components of the plant cells can include a cell wall, chloroplast, mitochondria, nucleus, so on. And what you can also include is that animal cells have a plasma membrane, they have organelles, they have nucleus, nucleolus, mitochondria, and so on. And you can say the difference between plant and animal cells could be the cell wall, plasma membrane, and so on. So just a few sentences introducing plant cells and animal cells will be great for this introduction. Okay? And your hypothesis. For this particular lab, there is no hypothesis. So you can write on these lines, no hypothesis required. This is an observation lab. Okay, this is an observation lab. Now let's go to the next section, which is your materials. In a neat column or a neat list, you want to list the materials that we use to carry out our lab today. So we used a compound microscope. We used prepared slides on the apical root tip, and I think the other one was the epithelial plant cell. We also used slides, slide covers, toothpicks, and we used blue food coloring. We should have used methylene blue, but I didn't have the methylene blue dye handy, so you can note that in your materials. Okay, next you're going to write down the procedure. Well, you can say that the plant slides, the two plant slides that we observed were pre-prepared slides, and you observed them in the microscope at 40 magnification, 100 magnification, and 400 magnification. And you would write that as 40x, 100x, 400x. So that tells us how far we zoomed in. The second part of your procedure was how we obtained the human cheek cells. You used a Q-tip and gently scratched or rubbed it against the inside of your cheek, and we made two specific kinds of slides. The first one was a dry mount slide where we just used that that toothpick that had the cells, and we swiped it against the slide and placed a slide cover on top of it. That was the dry mount. The wet mount was the same procedure, but then we added drops of the blue food coloring. So we made a dry mount slide and a wet mount slide of the human epithelial cheek cells. So you want to list your procedure in this section. If you need to add more paper, that is fine. If you need to change the format of this document, that is fine too. I will provide you with this document electronically so you can tailor it to your needs. Now, results should include any information or any observations, any drawings. So since this was an observation, I would expect you to have some really good pictures that you have drawn of what you observed. Now, the conclusion and discussion will just be that you were able to learn how to properly use and handle a microscope, how you were properly able to view pre-prepared slides, and how you were able to successfully make a dry mount and wet mount slide for viewing. The other part of this lab is going to be your drawings. So your drawings should be as detailed as possible. And you should also include a title of what you were actually drawing. You should also include the magnification in which you were viewing the particular specimens. So you can use the sheets were given to you in class to complete your drawings, or you can make your own sheet. That's totally up to you. And when you're doing this drawing, you can even color it in. If what you saw was in color, you can draw it and sketch it in color as well, okay? So this is going to be really exciting for me to see your very first lab reports. But all of the sections that were listed must be included in a lab report. Thank you so much for taking the time to do this lab report. I'm looking forward to reading it. Have a great day. Thank you.